Yeah, so our second speaker is Dr. Nick Venter. Dr. Nick is a researcher in the School of Animal, Plant, and Environmental Sciences at the University of Wits, specializing in biological control of invasive alien plants. His work focuses on discovery, evaluation, and implementation of insect-based solutions to support sustainable management of invasive weeds in South Africa. Today, Dr. Nick will take us through the current status of Bugweed biological control program. Dr. Nick, the screen is all yours.
Thanks, Sella. Thanks for having me. Okay, thanks. So following up from what Terry said, so at Wits, we've been at this for about 10 years now, following on from Terry, and yeah, it's bedeviled us as well. It's a very frustrating target plant to work on, and does pose a lot of challenges. But I'll go through some of these in the talk and some of the options that we have. And some of it will be reinforcing what Terry said, but just maybe using a different way of explaining it. And yeah, let me get on with it. So why do we use biological control? Well, I mean, Terry has alluded to why it's important, but it does also align with various developmental goals, UN developmental goals, such as clean water and sanitation, particularly for your aquatic weeds, responsible consumption and production. So biological control is a method you can use and be less reliant on herbicides, which can be detrimental to the environment and life on land. So clearing up the land and having less competition for native plants and agriculture. So yes, bugweed has been a challenge, and this is largely due to the fact that it is closely related to important agricultural species. So the solanums, potatoes, tomatoes, and eggplant, and other solanaceous crops such as chilies and peppers. Our work has shown in particular that eggplant, not the fruit itself, but the leaves and the flowers, are quite susceptible to non-target feeding from agents we have tested, and potato leaves as well. So Terry has explained how biocontrol works, but I can't help myself trying to explain it to the audience in a more visual manner. So what is biological control or classical biological control? So if you think of a pest that has been introduced into South Africa or Africa at large, Terry spoke of a lot of our pests come from Australia, but a lot of them do come from South America. And this is largely because we share similar latitudes. Shared similar latitudes do mean similar climates. And similar climates are obviously beneficial to these plants, and it does facilitate invasion. So the example here, water hyacinth, introduced into South Africa many years ago. Well, then as a researcher, when it becomes problematic, jump in the aeroplane or boat, depending how you like to be transported, go back to the native range and do surveys on the plants looking for insects. And using your experience and skills, you try and look for insects that may only be found on those plants. So you look at plants that are occurring adjacent to them or other closely related plants in that environment. And then with all the relevant permits, as we've spoken about, you bundle them back and you bring them back to South Africa, and then you start doing your testing. And it can take a few years to many years, but hopefully at the end of it, you can release a biological control agent. In an example, yeah, it's one of the weevils near Katana that is a biocontrol agent on water hyacinth. So what do we hope to achieve from classical biological control? So for all the university students out there, and some of you may still remember this, it's a classic predator-prey relationship. So what you're trying to achieve is not full eradication. I mean, that is a great scenario and it has happened, but ultimately you just want to reduce the population of the pest. Just apply some top-down pressure and get it to a manageable level. So as you can see, you've got the pest relationship at the top there, the red line, and then it's really high. So what you do is introduce the agent. Once you have tested the agent, the idea is once you introduce the agent, you can bring the population of the pest down, and the agent population will obviously increase concomitantly. And at some point, you should reach a new equilibrium or a stable state, as is seen in most natural systems. Because these plants are introduced into a novel range in the absence of their natural enemies, which is the premise for why weeds tend to become problematic. and you want to reintroduce them with their natural enemies and ultimately achieve a new equilibrium stable state. That's the dream. However, it's not always the reality. So we've heard a lot about Gargaphia decorus, and we've done a lot of work at WITS, Blair Cowie and myself on bugweed. And we've really wanted to see what is the impact of these agents on these plants, because not much work can be done on the direct impact on the plants. We're curious to see, can they be damaging? What we found using various experiments is that the lace bug in this example is very effective. So as mentioned, it causes chlorosis, which significantly suppresses photosynthesis. What this graph is showing you is that it also suppresses transpiration. Now, transpiration is evaporative loss of water through leaves, through the stomata. So not only is it suppressing the plant, suppressing its growth, suppressing fruit production, it also causes the plant to use less water, which is really important, particularly in water-stressed country like South Africa and elsewhere in Africa. So the secondary effects of its herbivory are really important, and it has the potential to be really damaging at a plant level. But as mentioned, its establishment has not been that great, and it's not very clear yet, but these hatched areas are essentially its potential establishment range. So it is more tolerant to cooler climates. And this establishment range basically encompasses the range of bugweed. However, as mentioned again, it is susceptible to predators. And one of the other things is potentially when it gets into the more high-fault region, above 1,000 meters, into the cooler areas of Gauteng, the plant quality does decline significantly. In fact, I mean, anybody that's in Gauteng, when you see bugweed in winter that aren't in gardens or being well watered, they look very sad. And this potentially has an impact on the Gargaphia decorus. This is still something I want to investigate, looking at the change in plant quality and see if this has a direct impact or subtle indirect impact on the Gargaphia. Similarly, Anthinomus santacruzi, the flower-feeding weevil. So in the field, it is really difficult to measure its impact. So it was mentioned that does seem to be a reduction in the number of fruits in the field. But that is a very difficult thing to test in the field because you have no control, and it's often hard to show causation if you don't have a control. So we know it should impact the reproductive output of these fruit because they're oviposit in the flowers. Also, the adults eat the anthers of the bugweed flowers, significantly affecting the ability to produce pollen. So from the experimental stuff we've done, we did show that the direct effects of Anthinomus adult feeding and larval feeding on the fruit was a 66% reduction in fruit set and about a 15% reduction in seed set. So the direct effect is a big reduction in fruit and a big reduction in seeds. And the indirect effects of the feeding were increased self-pollination. So as they crawl around the flowers, they're inadvertently self-pollinating the flowers, which then also reduces the viability of the seeds. So overall, they're showing a reduction in fruit set, a reduction in seed set, and of the seeds that are remaining, there is a decrease in seed viability. So as it stands, well, as a biocontrol agent, they do have the impact or the potential to be really impactful. However, this is not always realized in the field, and this is largely because bugweed produces so many fruit, and they are able to self-pollinate, so they are able to, well, not self-pollinate, but they're able to produce seeds without a pollinator, and they're also well pollinated by native honeybees and carpenter bees, so they are well positioned to produce good quality fruit. And again, climate is a big constraining factor for Anthinomus. So the hatched area just along the coastline here indicates their potential establishment, and it goes a little bit inland here up to Pietermaritzburg. So they are severely constrained by climate. So anywhere beyond this area into the high-fault here from 1,000 meters, it just gets too cold. And the question was controlled in the high-fault. We have tried on numerous occasions to release Gargaphia under very controlled conditions and protected conditions by bagging them, putting sticky tack on the stems to prevent ants, We've done similar things with Anthinomus, and we have not been able to get them to establish in Johannesburg. It is just too cold, and I think largely for the Anthinomus, the plant quality is really poor. It's not to say we should give up, because it does take a lot of resources and energy, but it doesn't happen very easily. So climate has been a large constraint to the establishment of Anthinomus. So overall, these red squares on top of the gray squares indicate where agents have established. So from Terry's previous talk, from his talk, only these areas here had showed establishment. We have discovered establishment down towards East London, but not further than that. So where they are established, they are abundant. So if you find a bugweed, pick a few flowers, you're very likely to find a weevil. That's always rewarding because you know they have dispersed. But they have not gone any further into these areas where the bugweed is persisting. So essentially, where this blue shaded area is, we cannot get establishment of any of these agents. And other work we have done has shown that pre-2000, the dark purple squares shows the distribution of bugweed. However, post-2000 to now, or to 2024, bugweed has expanded its range. So it is moving further inland and increasing its density along the coast. So its spread is increasing, and this obviously necessitates that we look into another agent. So when we were still funded, we started looking in areas where we thought we might find more climatically suitable agents. So myself and Blair and students of ours have made numerous trips to Uruguay in South America, because Uruguay shares a much similar latitude to us than areas of Argentina and Brazil. So the hypothesis is that we should be able to find agents that are more tolerant to cooler climates, particularly those found in the higher inland areas of South Africa. So we've done a lot of sampling east of Montevideo, particularly around Punta del Este and a little village called Agua. This area is a relatively high rainfall area, and there is a lot of forestry there as well, pine and eucalyptus forestry. And the most promising agent that we brought back is also an Anthinomus, but this is Anthinomus morticinus. So very similar to Anthinomus santacruzi, just a teeny tiny bit bigger. Not very noticeable, but for the trained eyes, when you work with them a lot, you can see they're slightly bigger. And we have bought, about 6 years ago, I think we started in 2019 with this, brought them back into the Wits insectary to start some host range testing. Anyone knows Joburg recognizes Braamfontein. So this is Jan Smuts Avenue along here. And we have started testing these agents. We have done the majority of testing, and we're testing them on various native solanums. I think that's tomentosa, Solanum tomentosa, and Solanum lichtensteinia. And we have also tested them on agricultural solanums. But what was really interesting is we wanted to test their thermal tolerance. Now, bear in mind, this is all done in a quarantine environment because these have not ever been approved for release. So we can look at the critical thermal minimums, so thermal physiology of the established weevil that Terry released, and it has a CT min, or critical thermal minimum, of about 4.7 degrees. So that's basically the temperature where they start losing physiological function, such as coordinated movement. And we tested the imported candidate weevil, and it had a much lower critical thermal minimum. So about 3 degrees low, which is very promising. It means that we have the potential for this agent to now start moving further inland and establishing in areas where the current weevil is not establishing. However, we have not released this agent yet, and we're still pending quite a few decisions. So as it stands, this agent is not being released. Largely because I think over time we've become slightly more risk averse, and by and large, the flower-feeding weevil's impact is relatively trivial. So we need to think long and hard about what we want to do with this current weevil. So as mentioned in the previous talk, I think our best option is to optimize current agents. And this strategy can be really effective. And an example here is anyone who knows about Hartebeespoort Dam in Northwest Province, so north of Johannesburg. Water hyacinth is a big problem in this dam. Most of you are familiar with it, and it's always in the news. So one of the more recent agents, a plant hopper, Megamelus scutularis, has been very effective, but this is largely due to the fact that research from the Center for Biological Control at Rhodes University started adopting the augmentative biocontrol approach, where they start doing inundative releases. So to get on top of it, you can see here, this is in spring. So the plant starts growing, but in reality, it takes the insect a lot longer to start increasing its population size. It's quite a long lag phase for the insect densities to increase to that of the plant density. So the idea is doing mass releases of these agents early in the season to try and bolster the population size to get on top of the plant population. So you can see here, this is in thousands of insects. So in the early part, spring, do big releases of these agents. And the data they collected beautifully illustrates these predator-prey relationships. So this solid line is the cover of water hyacinth on Hartbeespoort Dam. And then they do the releases of the agents. So as the population of the agents increases, we can see a nice decline in water hyacinth cover. And it's not because it's winter. You can see this is over the summer period. And we have this year in, year out here, this beautiful relationship where the inundative releases get on top of the water hyacinth and start reducing the cover. If anyone who follows the story knows it's not that simple, and there are a lot of other factors at play, pollution, politics, and so forth, but inundative releases are very effective. So it is an approach that has been recommended, and I often get contacted by the forestry sector asking what can we do about bugweed in the forestry sector. And my answer is always we need to mass rear Gargaphia decoris and do inundative mass releases in these environments, but not once and then walk away and rub your hands together and say, job well done. This needs to be done on a regular basis because of the susceptibility to predators and their outbreak nature, where they require certain favorable conditions for them to for their populations to explode. This needs to be done on a regular basis. And I think this is a strategy that needs to be adopted. The challenges in finding new biocontrol agents is very difficult, and we are not currently being funded to do this. But I think there is a lot of potential for the agents we do have. Experimental work and field work has shown that they are very effective at suppressing these plants, but we need to get them to really high numbers, like that seen at Hartbeespoort, where they do mass releases of tens of thousands of the plant hoppers on the water hyacinth on a regular basis. We need to do the same with Gargaphia decoris because, as mentioned, they are quite easy to rear as opposed to the flower-feeding weevils, which are a little bit more tricky and require a little bit more finesse to rear. And if we can do this, I think we can get on top of it. We can use this as a form of natural herbicide. So the biological control, continual releases of this agent. So we are still looking at investigating new candidate agents, and this is largely done through Landcare Research in New Zealand, which is now also the Bioeconomy Science Institute. So we currently are funded to assist with exploring new biocontrol agents for them. We have tested the morticinis for them. They only have 2 native Solanum species. There were subtle non-target effects. They are extremely risk averse, so we decided not to go ahead with that. But we are looking at revisiting some old agents and maybe looking for fungal pathogens, so pathogenic agents as well. So work is ongoing, and naturally anything we will find in through the work we do with them, we will obviously use and try and test against, use them to do testing in South Africa, and hopefully they can also be reused to be released against bugweed in South Africa. Okay, thanks very much. And if you're wondering, this photo is not from South Africa, this is from Uruguay, and plants that tend to be weedy here tend to be relatively weedy in their native range. So even in the eucalyptus and palm plantations in Uruguay, even though they are natural enemies, bugweed does tend to be quite weedy. So these plants have inherent traits, weedy traits. So that just makes it that much more difficult to control some of these weeds. Thanks very much.
Nick, thank you so much for that talk. Yeah, absolutely fascinating and a really great continuation of Terry's stories.
Yeah.
But while we were preparing for this event, you mentioned it's the Queen of the Nights.
Yes.
Okay,
Queen of the
so
Night cats.
the reserve that this event was actually planned for, the Klip Riversberg Natural Reserve, they have a problem with Queen of the Nights. And I was just wondering, you mentioned that in that case, biocontrol had been very successful. It would just be interesting to me to know why for that species.
So while it's working for that species,
Yeah.
okay. So it's got to do with the life history and the lifestyle of these species. The biocontrol agent for Queen of the Night, it's from South America. I'm not entirely, I can't remember exactly where in South America, but it has a very different life history and a life cycle. And it forms galls, essentially forms galls on the plant. So it is a scale insect, um, similar-ish to cochineal, but it creates a little microclimate within the gall on the Queen of the Night. So climate and cooler temperatures tend to be less damaging or less impactful on an agent like that. And then some insects just inherently have more cold tolerance than others, and also depends where they're from. So the same insect collected further south in, say, South America will inherently have a better thermal tolerance, cooler thermal tolerance than an insect collected further north. So there are population differences, but yes, that one essentially forms a gall and protects itself.
I see there is a question from David Harding. David is asking what studies have been done on the impact of these agents on the local food chain. Surely releasing so many agents has an effect on more than just the plants they are targeting.
No, there is no, I can say with surety, there is no impact beyond the plants that they are targeting. So these are monophagous insects. They are insects that have evolved a very close relationship with that species, or maybe a few species within a genus. So that plant has evolved over millennia to only be able to feed or complete its life cycle on a specific plant. Plants carry a lot of secondary compounds, secondary metabolites, and not every insect can cope with these secondary metabolites. In the absence of its host plant, so say for example, this biocontrol agent is really effective and it kills a host plant, which has happened, the agents will die. They cannot move on to another diet. And the counter argument for that is if they were to jump ship and start eating some closely related plants, why have our native species, of which there are tens of thousands of species, why hasn't at least not one started feeding on these invasive plants? You'll get some stray feeding by locusts and grasshoppers and that, but there are no specialized insects that have started eating our invasive plants. So nothing has co-evolved to be able to cope with them as a diet.
Okay, that wasn't my question. I know about host specificity. My question is, you are releasing tens of millions of insects, and I'm going to use the water hyacinth dams example. There are videos going around where people they can't brine more because there's so many insects in at their house. So we know when we have invasive plants, the one thing they do is they cause a change in feeding habits of indigenous animals. So now with all these billions upon billions of insects to the point where people can't open their windows, surely that's changed the feeding habits of something. Has that ever been studied? And is that something that we should be thinking about?
Okay, so that is, I mean, yeah, I've seen the videos. I'm aware of the explosion of these insects. It is quite unique to that situation, to Hartebeest. That is not something that you would normally find in any other biocontrol situation. And that's largely got to do with the inundative releases, these mass releases. But what is happening there is that the agents are so effective that they are it's depending in the situation, but often they have exhausted their food supply of the water hyacinth, and they're now trying to disperse to find more food. Naturally, they can only feed on water hyacinth, so they are dispersing. I think it's such a these events are so far and few between. I don't know if it has any long-lasting impacts. Yeah, it may impact the food chain for a short while, but these events are very short-lived. I mean, it could be studied, but very difficult to study because it is a very rare event. It's collateral damage. As biocontrollers, we find it fascinating to see these agents explode like that, and what we'd like to see, but obviously not with the amount of annoyance and grumpiness that has come from residents of Hartebeest with the explosion of these insects at those occasions.
It's actually quite concerning because it has to have an impact. That number of insects where people can't open their windows. I know you say
Yeah.
it's great because we're getting the water hyacinths done, but surely that's changed the impact of the feeding habits of the birds, of other insects. Like I used an example at the CBC talk a while ago. No one answered me. Like something as simple as, what are the dragonflies feeding on now? If they have such an abundance of something like the weevils and that, are they no longer eating mosquitoes? Is that going to suddenly cause an increase in malaria? I know this is a really out there topic, but these things need to be addressed. We can't just say they've run out of things to feed on, so now they're going to houses. There has to be a food chain linked. I think it's kind of irresponsible to just say, oh, look, they cause an annoyance to homeowners. They're not. That amount of biomass has to be causing downstream things. And I think it should be addressed and not just brushed off as like oh, well, it's a once-off thing. At what point does a once-off thing become an ecological disaster? That's what I'm getting at.
Well, I mean, to counter argue that the pollution in Hartebeest is an ecological disaster. It is a highly disturbed environment. So, yes, an outbreak of insects, there are naturally cascading effects. I'm not going to argue that. But we have a very disturbed environment where these things are happening. So this is not happening in a pristine setting in, say, Kruger National Park, where you would expect big effects. The whole situation, the whole environment is so unstable and so disturbed that if there is a measurable impact, is there actually a significant impact on the surrounding flora and fauna? But I don't say it's not a good point. The impact on eating mosquitoes is a good, valid point. I will defer my question to the researchers at the CBC because they are constantly faced with these type of questions. It has somewhat made me regret using water hyacinth as an example because it is a contentious issue. But my feeling is I'd rather do something than not do anything at all because everybody has an opinion of how these plants should be controlled mechanically. And there are secondary effects. So you remove the water hyacinth. Now we have a secondary invasion of Salvinia minima and the blue-green algae. This is largely and I will say it is largely a product of poor governance, but that's as much as I'm going to say about that situation. Thanks, Dave.
Okay. Thank you so much, Dave. We have another question from Natari. Natari says, how long does it take to mass rear a weevil population for release?
So the weevil, we streamlined the rearing of the weevils in our quarantine facility. For the first few years, we used quite a labor-intensive method, but then we learned to streamline it. You can build the population up quite quickly, but it does involve transferring buds to containers and so forth, having bouquets in that, because using living plants in pots don't produce enough flowers to produce enough weevils for mass rearing. That's why the Gargaphia is a better solution. So it could take many months to produce a few thousand weevils. So relative to Gargaphia, it isn't as effective, but it can be done depending how much effort you're prepared to put in. It's not quite as good as the the example of the plant hopper and water hyacinth that breeds up nicely. The weevils aren't as fecund as those plant hoppers. So yeah, with a little bit of effort, you can do it.
Okay, thank you. And there is another question from Dion. Dion is saying that here in a small community in Mpumalanga, we are dealing with a large infestation of bugweed. As a community, we're getting together this weekend to cut out, apply the herbicide caput to the stems, and to snip seeds off separately. How do we destroy the seeds on their own?
So you mean destroying the seeds once you've harvested them?
Yeah.
The best bet is to pile them up in a very secluded area and wait for them to die off. You could potentially burn it thereafter once it's dried out. If you're doing a stump treatment once you've cut the trees down. I think that's a case-specific situation, depending on the environment you're in and what you need to do. And unfortunately, because the biocontrol has not been effective, largely effective on bugweed, you're going to have to resort to mechanical and herbicidal treatment and just ensure that it is. I am not an expert on herbicides, but just ensure that it is a registered herbicide for the bugweed. I can't remember if kaput is. Thanks.
Okay. Thank you so much, Dr. Nick. And we're going to get our last question from John Simkonda. I think he's from the Department of Wildlife in Malawi, if not mistaken. John Simkonda, over to you.
Yes, in fact, I'll just share the link. I'm Edson from Nyika National Park, Malawi.
Ah,
Yeah. In fact, I was not part of the workshop, but I was just shared by my friend from Vaza, Malawi.
okay.
Okay, we've been working on invasive and alien species in Nyika National Park, especially at the plateau, with the wheat. I think for some years, I think for 5 years, I think in the past. But now we are looking at what are the possible measures to control, let's say, black and fin, black wattle, in pine, blue gum, and so on and so forth. Now we've had problems. We were using mechanical and as well as the chemical. We used chemicals where it was working in rubus and the and the other species like black wattle. But now the challenge we have here, I don't know if our researchers have done any kind of research regards biological methods, which I've just heard from Nick. Is on the black and fin, because the black and fin is causing a lot of problems on the plateau. So what we're doing now is just mechanical. We're just cutting and at times applying lime and so on and so forth. I don't know if biological methods have been tested elsewhere, and if it is working, could you advise us so that we can probably link up with those who have been working on this. Secondly, I was looking at the seed power bank from Pinus Patura. When you harvest pine, you find that you create an inducive environment for the growth of for the generation of the seed bank on the soil. So I don't know whether what would be the better method in getting away with these seed banks. That's my question. I submit.
There's not much you can do with the seed bank once it's in the ground, unless a really, really, really hot fire comes through, which can scorch the seeds and reduce their viability. But ultimately, you need a seed feeding biological control agent, something like is found on the wattles. But it does take very long for the effects to be seen in the environment. For the seed rain to be reduced, it does take very long. So it is quite a long-term gain. So once it's in the ground, there's not much you can do. Finding a biocontrol agent that will then feed on seeds in the seed bank is very rare. That would normally do it on a plant. So that's a situation where you'd have to continually go back and mechanically remove or spray germinating wattles, for that example. But other than a seed feeding agent, there's not much you can do about the seed bank. It's just a game you got to wait. For example, water hyacinth, the seeds can live for 20 years in the mud underwater. So also extremely long-lived seed bank. And you just got to let that seed bank work itself through. And that's with the applied control. Eventually, if you can suppress the flowering of the plants, the seed bank eventually will deplete itself. But for any other biocontrol, you're more than welcome to drop me an email, and where I can help, I can direct it to the right people. I know Center for Biological Control is doing a lot of work in Africa, But it's not as simple as just releasing an agent, because if we were to release the biocontrol agent for bugweed in Malawi, we would have to do some host range testing on your native Solanum species. So it's not a matter of it's been tested yet, it's safe for anywhere else in Africa. That's irresponsible. We would have to test its host range on your native Solanum plants, which does complicate the matter and does add cost and time to the project, but there are ways of doing it.
Okay. Thank you so much, Dr. Nick.